Hi everyone, today we are going to continue, and this should be our final part on energy and enzymes. So we are going to learn today about what could affect enzymes, factors that affect enzymes. So let's begin. So factors affecting enzyme function could include things like enzyme concentration. So how much enzyme is present in the cell or in the solution is going to affect how the enzyme works. Substrate concentration, how much substrate we're putting in the cell or the reaction. The temperature can affect the function, the pH, and there are things called active and things called inhibitors. These both affect enzyme function as well. So we're going to begin by talking about enzyme concentration. So enzyme concentration can affect a reaction in the following ways. So let's take a look. We see the enzyme concentration increases as we go to the right, the reaction rate goes up, and we see that as enzyme concentration increases, what happens to the reaction rate? The reaction rate increases as well. But what do you notice? It seems like it should be a straight line, right? More enzyme, faster reaction. We see that it slows down and eventually comes to a plateau. So what could be causing that plateau? Take a minute and think about what might be causing a plateau like that. Well, let's think about what's happening. The enzyme concentration goes up. At the beginning, the reaction rate is going to go up, right? Because if we think about it, we have more enzyme, the reaction is going to happen faster. Move this stuff out of the way. But notice what happens. So more enzymes, they will more frequently collide with something. But what happens is the reaction rate off is that eventually we just start running out of substrate. There's a certain amount of substrate. If we have, 1,000 substrate molecules and you're putting in 2,000, 3,000, 4,000 enzymes, well, you can put in as many enzymes as you want, but they won't have anything to work on, right? So at some point, right around here, we start having too many enzymes for the amount of substrate that we have. And that's shown in the picture. There is some unbonded substrate, but there's not that much. We have way more enzyme than we need. And that is why the reaction starts to level off. Not all enzyme molecules can find substrate. Okay, substrate concentration. Look, it's the same graph. So our penguin is again going to ask, what's happening here? Take a moment and think about what is going on. Well, we have come to this conclusion, but it's the same as with the enzyme, right? When we add more substrate, we're increasing the reaction. More substrate means that the substrate is more frequently going to collide with the enzyme. But the reaction rate eventually levels off because at some point, like we see in the picture right around here on the graph, all of the enzyme is pretty much full. There's very few enzymes not taken. So we end up with a lot of substrate that just doesn't have any enzyme to bind to. At that point, the enzyme is saturated. And when the enzyme is saturated, we see that plateau starting. So we've reached the maximum rate of reaction. Now, what about temperature? Temperature graph is going to look something like this. Okay, so what's happening around 38 degrees Celsius? Well, that seems to be the ideal temperature for the enzymes to work. Notice the enzymes don't work very well at very cool temperatures, and they start to work better as the temperature goes up. There's an optimum temperature, and then when the temperature gets too hot, the enzymes are not able to work as well, and eventually they stop working because they are denatured. So down here, we have a denaturing of the enzymes. Over here, the cold doesn't denature the enzymes, but because when things are colder, they move more slowly. There is just not enough collision between enzymes and substrates to really cause any sort of reaction. Coldness slows down the molecules, so they can't collide. The heat will speed them up, but it will also denature the enzymes. All right, so here we have temperature. There is an optimum temperature, and that is when we have the highest number of molecular collisions. In humans, that's going to be between 35 and 40 degrees Celsius, or normal human body temperature. If heat goes beyond the optimum, the increased energy level of the molecules disrupts the bonds in the enzyme and between the enzyme and the substrate. So we have denaturation. And when it's too cold, the molecules simply move too slow. So we have a decrease in collisions between the enzyme and the substrate. Different enzymes function in different organisms that live in different environments. So in this graph, we have an optimum temperature of 37 for humans, but things, the optimum might be higher. For example, so here we have, let's move this out of the way, we have 158 Fahrenheit, if you can see that there. And this is for prokaryotes that might live in a hot spring. If they're living in a hot spring, the optimum temperature for their enzymes is going to evolve to be hotter, which it has. All right, different organisms are going to have different mechanisms for dealing with this. So animals like mammals that we call endotherms, these are animals that maintain a body temperature regardless of whether we're cold or we're hot. We have these mechanisms to maintain homeostasis over time. So the bobcat there keeps its temperature the same for the sake of its enzymes. A snake is an ectotherm. So a snake can actually tolerate a wider range of temperatures, and its enzymes can usually tolerate a wider range of enzymes. But if it gets too hot or too cold, usually a snake will try to find a place to cool off or warm up by changing its location. And some animals that are ectotherms may go into like a type of hibernation, depending on if it's too hot or if it's too cold, because their enzymes won't be able to work very efficiently. Is another thing that can change enzymes. Most living things are going to be around a pH of 7, so that is the ideal pH for their enzymes. However, and pepsin, which is found in your stomach, has a lower ideal pH. Why might that be? Well, it might have a lower ideal pH because your stomach is filled with hydrochloric acid, which is acidic. Sometimes the ideal pH might be basic, like trypsin. Trypsin is in our small intestines, and our small intestines are a little bit basic. That's because food moves from our acidic stomach, which is equipped to handle the acidity. It moves into our small intestine, and our small intestine is not equipped to handle acidity. So our pancreas pumps some sodium bicarbonate in there as like a buffer, and that makes it slightly basic to counteract the acidity so that our intestines don't get destroyed by all the acid from the stomach. All right, so that is what's going on here. pH changes in pH are going to add or remove hydrogen ions. And as we know, that 3D shape of proteins and enzymes, that's very delicate. Adding or removing hydrogen ions is going to disrupt the ionic and the hydrogen and all of those bonds that cause that three-dimensional shape. Adding or removing hydrogen ions due to changing acidity is going to disrupt. It can affect the secondary and the tertiary structure, which may be the protein. An optimal pH, again, most humans' optimal pH is around 7. But on a localized condition. Pepsin and trypsin are two examples that are different. Okay, salinity is also going to have a certain amount that is ideal. Too much or too little salt, that's going to affect things. The salt concentration can basically is going to add or remove cations and anions, which is going to disrupt many of those ionic and other bonds that are holding together the dimensional shape. It's going to disrupt the attractions between charged amino acids and therefore to cause denaturation. So many enzymes are intolerant of extreme salinity, like the Dead Sea. It's called the Dead Sea because very few living things can actually survive in such a high salinity area. Okay, there are some other things that may affect enzyme function. They are known as cofactors or coenzymes, and we'll be watching a video about this. So Compounds which may help an enzyme work are called activators, and cofactors can act as activators. Cofactors are non-proteins, so they may be inorganic compounds, and they may even be minerals that help an enzyme do its job. Magnesium, potassium, calcium, zinc, iron, and copper are cofactors. If we look at hemoglobin here, hemoglobin has a cofactor of iron. This is chlorophyll. Chlorophyll has magnesium. These are minerals that help enzymes do their job. So they are bound within the enzyme, and they are directly involved in the action of the enzyme. So iron and hemoglobin, magnesium, and chlorophyll, these are cofactors. Coenzymes are non-proteins, but they're organic. So they're not proteins, but they're still organic molecules. And they can bind temporarily or permanently to the active site or near the active site of an enzyme. Vitamins are organic, but not proteins. They are coenzymes. So niacin, B3, they're going to help our electron carrier, NAD+, carry electrons. Riboflavin, B2, that is also going to help one of our electron carriers, FAD+, carry electrons. That's another one of our electron carriers that we've learned about. Coenzyme A is going to carry acetyl groups. That's also going to be important for cell respiration. Okay, so sometimes it is necessary to limit. So we talked about things that activate an enzyme. What about things that limit enzyme? There are things that are called inhibitors, and inhibitors are going to reduce the activity of enzymes. Okay. Sometimes that's important, right? Sometimes we want enzymes to stop working. It's in order to maintain homeostasis, we need enzymes to work when necessary. It also means we need to stop enzymes from working too much, right? We don't want all of them to go all the time. So there is competitive inhibition, non-competitive inhibition, and sometimes inhibition is irreversible, and we're going to talk about that. There's also feedback. We'll get to that in a minute. So competitive inhibitor is going to compete for the active site. So we see that in the picture over here. The enzyme should fit with the substrate, but there is a competitive inhibitor that is blocking the substrate from binding. So this is a way that a living thing can turn an enzyme off. They can use a competitive inhibitor to stop a substrate from binding. Perhaps we have too much of a product, so the body says, make this particular inhibitor so that we don't get any more of this product. We want to maintain homeostasis. Penicillin is used to block an enzyme that bacteria use to build cell walls. So this is not something that bacteria are going to make themselves. This is something that we use to inhibit bacteria from growing and surviving. So penicillin is a competitive. We can treat chronic alcoholism. So we use a certain chemical that's going to block an enzyme that will break down a byproduct of alcohol. That's going to make things very unpleasant. If an alcoholic on this medication does drink alcohol, they're going to be in a lot of pain because this byproduct doesn't get broken down the way that it should. So they kind of associate alcohol with this horrible, horrible feeling caused by the byproduct. So it's going to cause a lot of very negative effects very soon after drinking. We can always overcome, well, we can usually overcome competitive inhibition by increasing the substrate competition. This is going to outcompete the inhibitor, right? So, for example, methanol is a type of alcohol called wood alcohol that people wouldn't normally drink. If you do drink it, it can make a particular chemical that's going to damage your optic nerve, which can cause blindness. So if you have methanol poisoning, then you might treat that by taking in some ethanol. The ethanol then will outcompete the methanol, and ethanol is preferred. Then we have non-competitive inhibition. Non-competitive inhibition is sometimes called allosteric inhibition. And that's because we are working with an allosteric site, which is another part of the enzyme. We can see the active site on top. The allosteric site is this other cookie cutter shape. So it is a site that's not the active site. An allosteric inhibitor is going to bind to the allosteric site, and it is going to cause the enzyme to undergo a conformational change. So if we take a look at the picture on the right, this enzyme has an allosteric inhibitor bound to the allosteric site, and that has caused a conformational change, and now the substrate no longer fits in. So we call it non-competitive because it's not competing for the active site, it's actually going around the back and blocking it from this other tricky little way. So that is a way of making sure the active site doesn't have the correct shape. It's a way of turning the enzyme off. Some anti-cancer drugs work this way. So cancer is going to cause this very fast cell division. So you might have a non-competitive inhibitor, an allosteric inhibitor that attaches to an enzyme that's basically going to prevent DNA production. If we can't produce DNA, we can't divide our cells. So it's a way of stopping cancer cells from dividing by non-competitively blocking an enzyme. Cyanide poisoning works in the same way. It's going to stop an enzyme that helps produce ATP called cytochrome C. And cyanide poisoning is going to work in, like we see on the right, a non-competitive way. It's going to block ATP production, which of course we need ATP for energy. Some types of inhibition are actually irreversible. So that cyanide poisoning is a permanent bond. So that was non-competitive. So we can have irreversible competitive inhibition, and we can have irreversible allosteric inhibition. So things only bind to either the active site or to the allosteric site. Other non-competitive irreversible inhibitions would be things like nerve gas, so something like sarin or insecticides. These are called cholinesterase inhibitors, and what they do is they block the shape of the active site from doing its job, basically. So a neurotransmitter, acetylcholine, needs to get broken down. So acetylcholine is going to help your muscles move, and these things basically prevent it from being broken down. So it causes this constant contraction of muscles, which is very, very okay. There's also allosteric regulation. So we can turn on or turn off enzymes with the allosteric site. Conformational change caused by regulatory molecules that can inhibit or activate. So we see conformational changes are happening. Where is that? Due to allosteric regulation. Here we have an allosteric site that is filled with an activator. It's turning on the active site. And here we have an allosteric site with an inhibitor. It's turning off the active site. So it can do either. Allosteric sites can turn on enzymes. They can turn off inhibitors. Enzymes in the inactive form. Activators keep enzymes in the active form. We also have metabolic pathways. Let me move my head out of the way. So if we want to take a certain molecule and change it into another molecule, we may need a number of enzymes to do that. So we may need enzymes to undergo each of these steps. Look at all those enzymes. So if we divide that chemical reaction into many small steps, it becomes easier to regulate. So it may seem like it's not a great idea, but in fact, it does help things along in some ways. It's an artifact of evolution, and it increases efficiency because it allows us to control the process in any of these steps. So we see the substrate binding to enzyme 1, becoming something new, binding to enzyme 2, a third, binding to enzyme 3, and so on until we get our finished product. So this increases our control and allows us to regulate the production of the product. One example of this that we will go over in cell respiration is in the mitochondria. Mitochondria, we have these enzymes situated in the membrane for efficiency. These enzymes are in the correct order already. All right. And then we also regulate it by using feedback inhibition. So the product is used as the reactant in the next step of the pathway, and the final product can inhibit an early. So what does that mean? Well, it means that when we make product G can actually go and block enzyme 1. G can be an allosteric enzyme for enzyme 1. Why would we want that? We might want that because it prevents us from making too much of our product. In this case, product G. We may only want a certain amount. So the production of product G also turns off the pathway that makes it. The fantastic way that evolution has devised to make sure that we don't have too much. We have just enough of one product. When product G starts to get made more and more and more, it turns off its own pathway, which is a good thing. So that's feedback inhibition, and that means there's no unnecessary accumulation of the product. Ingenious thing. Here, we have an example of feedback inhibition. We have synthesis of the amino acid isoleucine from the amino acid threonine. So this is a metabolic pathway. We're taking one amino acid, we're turning it into another one. That's happening here. And that isoleucine turns off its own pathway, right? Prevent too much being made. So isoleucine becomes the allosteric inhibitor of the first pathway. As the product accumulates, it collides with the enzyme more often than the substrate does, and that turns it off. All right, we are going to stop there. Let me know if you have any questions. I know this was a long one, so we will be reviewing this soon.